Bismillahirrahmanirrahim. Today we are going to study about cell wall. Cell wall is a firm non-living layer that encloses and supports cells of most plants, bacteria and fungi. This term is first described by Robert Hooke when he discovered cell. It surrounds the plasma membrane of plant cells. It is an extracellular component that is present exterior to the plasma membrane. Functions of cell wall. It gives cell a definite shape and structure. It provides structural support to the cells in which it is present. It provides protection against infection and mechanical stress. It also provides protection to the plant against osmotic stress, and it helps to develop turgor pressure, which is the pressure of the cell contents against cell wall. It separates interior of cell from the external environment. Cell wall also enables transport of substances and information from cell inside to the exterior and vice versa. Now cell wall is present in plant cells, prokaryotes and fungi. It is secreted by the cell. It means that cell wall is derived from minute vesicles produced in Golgi complex. Golgi complex or Golgi apparatus is an organelle that is present in cytoplasm. It is a double membrane bound organelle. It produces minute vesicles. At the time of cell division, these vesicles assemble in the center of the two cells. When these vesicles fuse, they form a cell plate that divides the newly produced daughter cells. At that time, cell wall is formed. So we can say that cell wall is a secretory product of plant cell cytoplasm. Next, cell wall is a porous membrane that allows free passage of water molecules and dissolved material. It means that cell wall is also a permeable membrane. Now we talk about the composition of cell wall in different organisms. In plants, it is made up of cellulose. In prokaryotes, it is made up of peptidoglycan or murein, while in fungi, it is made up of chitin. Next comes the layers of cell wall. There are three main layers of cell wall: secondary cell wall, primary cell wall, and middle lamella. Sometimes we can also call middle lamella the tertiary cell wall. In this picture, we see the layers of cell wall. First of all, secondary cell wall is present. Next, primary cell wall. After that, middle lamella. These all layers are shown in different colors. If you see, then secondary cell wall is present in orange color, middle lamella in red color, and primary cell wall is shown in yellow color. So we can easily differentiate in this picture all the three layers of cell wall. Now we talk about primary cell wall. Primary cell wall, each cell produces a primary cell wall. It is a true wall. It develops in newly growing cells during cell division. It is present inner to middle lamella. It is thin and slightly flexible wall. Now microfibrils of primary cell wall show a criss-cross arrangement in layers one above the other. This feature provides the cell a great strength. Primary cell wall is also adapted to growth, and this wall stretches prostically or irreversibly. It means that primary cell wall is formed in newly growing cells, and when the size of cell increase, primary cell wall can also increase until the cell attains its maximum size. In this picture, we can see the criss-cross arrangement of microfibrils present in primary cell wall. Now we see the composition of primary cell wall. It is made up of cellulose microfibrils, bundles of cellulose chains, hemicelluloses, and pectin. In this picture, we can see middle lamina, primary cell wall, and plasma membrane. And on the next side, we see hemicelluloses, pectin, cellulose microfibrils, and soluble proteins. These all materials are shown in different colors so we can easily differentiate between hemicelluloses, pectin, and cellulose microfibrils. Now we see the secondary cell wall. It is present in sclerenchyma cells that are dead and provides spore to plants. It is formed between primary cell wall and plasma membrane. It develops only when cell has reached its maximum size. Secondary cell wall is deposited after cessation of cell expansion in specialized cell types, including tracheids, vessels, fibers, and sclerae. These all cells are collectively known as sclerenchyma. Secondary cell wall is most prominent in secondary xylem of gymnosperms. Secondary cell wall is a thick and rigid wall. It does not allow further growth once produced. In this picture, we see different layers of secondary wall. There are secondary wall S1, secondary wall S2, and secondary wall S3. Composition of secondary cell wall. It is made up of cellulose, hemicelluloses, lignin, inorganic salts, and waxes. In this picture, we can see cellulose, hemicellulose, lignin, and pectin. We can also see primary cell wall and secondary cell wall in this picture. Now, cellulose microfibrils also show a crisscross arrangement in secondary cell wall. Lignin, the other major component of secondary cell wall, it cements and anchors cellulose microfibrils together. It is mainly responsible for rigidness of secondary cell wall. This wall provides a definite shape and mechanical core to the cell. Middle lamella. It is present between adjacent primary cell walls of two cells. It is composed of sticky gel-like magnesium and calcium salts, pectic acids, and pectin. It holds neighboring cell walls together. Now pectin. Pectin is a polymer of around 200 lacturonic acid molecules. Majority of its carboxyl groups are methylated. It is less hydrophilic than pectic acid but soluble in hot water. Major component of middle lamella but also found in primary walls. Pectic acids. These are polymers of around 100 lacturonic acid molecules. They are very hydrophilic in nature. They form salts with calcium ion and magnesium ion that are insoluble gels. They are major components of middle lamella but also found in primary walls. So this is all about today's lecture.